So thank you, Lisa, for the kind introduction and for the invitation to speak with you today about my interest in measuring human auditory evoked potentials. So as Lisa said, my background is very different than yours. I come from the field of auditory neuroscience. I studied at McMaster University under the mentorship of Laurel Trainer. And I'm going to talk to you a little bit about my work there today, but I'm going to talk a little bit more about how this type of research can be applied to open up an exciting new frontier at Orca US, where we could learn more and better develop our understanding of the processing dynamics in the auditory pathway and how such dynamics might be affected in listeners with sensory neural hearing loss. So just to get us started on the same page, I'm sure that you can all agree that one of the most challenging tasks of the mammalian auditory system is the perception and discrimination of behaviorally relevant sounds, especially extracting such sounds from background noise. In bats, this can take on the form of echolocation calls. For mice and other rodents, this could be the detection of the sounds of a predator so that they can flee. But for modern humans, arguably the most important application of this system is in deciphering speech signals, those signals that form the core of our verbal communication, or auditory communication. So, from a purely acoustical standpoint, there isn't a great deal of difference from a P or a B, or from a K or a T. But from the perspective of language comprehension, such confusions are the difference between beak and peak, or beat and peat, and can have a profound effect on the efficacy of communication between two individuals. Accordingly, avoiding these confusions becomes even more difficult when trying to extract relevant signal from background noise, or extracting a particular speaker or a particular stream of signals from an environment where you have competing acoustic signals with overlapping spectral temporal features. And as you can imagine, when the signal itself is degraded, as is the case with sensory neural hearing loss, the task can quite literally become impossible for some people, which often leads to reports of feelings of social isolation and overall poor perceived quality of life. Indeed, even in good cases, sufferers of hearing loss can exhibit demonstrable deficits in speech and noise tasks, even after corrective amplification, and this can become very troublesome for both the patient and the clinician. Now, developing a better appreciation of this problem, in my opinion, has required quite a large shift in the way that we think about hearing and in how hearing science has approached the auditory system. And this is a shift that's happened mainly over the last 2 decades. It's one that's moved away from perceiving the auditory system as a passive sensory process where acoustic input stimulates the cochlea, that cochlear transduction turns the physical properties of the sound into a neural representation in both the rate of firing and the place of firing in auditory nerves. This neural representation is sent up through the nuclei of the subcortex until it reaches the thalamus and auditory cortices, where the features become integrated into auditory percepts. This is the normal hierarchical model of listening. This is the one that we've considered most often in the past when we think about hearing. But this framework is incomplete, and it's incomplete because it ignores, namely, this complex array of descending projections that go from auditory cortex and all of the subcortical nuclei and feed back down. These are some of the largest pathways in the brain, period. And for years and years, we've known very little about what they do and what their function is in hearing. Only recently has an appreciation of this circuitry recast the auditory system as one that is no longer a passive system of relays that increase their complexity of processing as they go up from subcortex to cortex, but rather one in which a series of dynamic loops allows ascendant stages of the auditory system to feed back and modulate the representation of certain sound features at preceding stages. So in this updated view, you can think of sensory information coming in, being encoded at the level of auditory subcortex or brainstem, This process is bottom-up, it's exogenous, it's driven by stimulus features, but then those stimulus features are passed up to AC, and I gave away the most interesting part of this, is that now we have this feedback loop here that we're just starting to appreciate, this so-called cortical fugal or top-down feedback that allows auditory cortex to fine-tune the incoming auditory information according to whatever central demands that are placed upon the auditory system. So if we return to the problem of trying to listen to your friend in a noisy restaurant, such a dynamic control system might see the brainstem structures encoding sensory information of your friend's voice, the harmonic content there. This harmonic content is then processed by auditory cortex, which extracts the meaningful relationships among those harmonics, the ones that actually define your friend's voice from a background noise. And if the situation is sufficiently noisy, the AC can, through these cortical fugal projections, these efferent pathways, actually boost the response at the certain harmonics that represent your friend's voice, and it can attenuate the responsivity of subcortical neurons to irrelevant acoustic features. So where might we start looking for these circuits in the brain? Well, here's one good place to start. These are the pathways that define the so-called colliculo-thalamo-cortical colliculo loop. Now, it's a mouthful. I'm going to focus on the reciprocal connections mainly between IC and AC. I've included the thalamus here, the medial geniculate. That's the auditory part of the thalamus because it's also involved in this. But a lot of the work that I'll be talking about today focuses mainly on tuning of response properties in inferior colliculus. So why look at the inferior colliculus? Well, it's a major convergence hub in the rostral brainstem for both ascending and descending auditory pathways. The central nucleus of the inferior colliculus is the target of nearly all bottom-up tonotopic projections. So those are projections that carry frequency information up from auditory nerve all the way through to cortex. And the IC is populated by a wide variety of neural cell types which respond to a wide variety of spectrotemporal properties in the sound. They can be cells that respond to single properties, intensity, frequency, or they can be cells that respond to a number of different properties and integrate those properties. So you have cells that respond to a particular combination of frequency and intensity, or even a combination of frequency and location cues. So the intraoral timing differences or intraoral intensity differences. Now, the dorsal nucleus of the IC, which is adjacent to the central nucleus, is the target of a lot of top-down projections from thalamus and from primary auditory cortex. Now, although this pathway is not tonotopic itself, so it does not actually, the neurons there don't actually respond in a tonotopic manner, they do communicate with neurons in central auditory, or in the central nucleus of the inferior colliculus. So these pathways that innervate dorsal cochlear nucleus can, in fact, have an effect on the ascending information from IC. And we do see that that is the case, at least in animal models. So Nabu Sugo and his colleagues have done a lot of this work, looking at what happens if you stimulate a particular part of auditory cortex that's tuned to a best frequency. And by best frequency, I simply mean the frequency at which the neuron's response threshold is lowest. So what is the frequency where you need minimal amount of intensity to still evoke a response? That's the best frequency. What happens when you stimulate a neuron in cortex with a particular best frequency to the tuning properties of neurons in IC? And if I didn't mention already, this is in the big brown bat. So 3 things can happen when you run this kind of experiment. The first is neurons in IC that share the best frequency of neurons in the auditory cortex, their tuning properties are actually enhanced. So they require now less energy at that particular frequency for an evoked response. This isn't an auditory evoked response. These are intracranial recordings now in the bat's auditory system. So you measure the neuron that's matched, it fires more vigorously now to the same level of stimulus. If you look at neurons that are unmatched but close to this matched region, their tuning properties shift. So now their best frequency isn't what was intrinsic at the beginning of the experiment, but it has moved over to be closer to the best frequency of the stimulated cortical region. In essence, cortex is recruiting more neurons to represent the particular sound that's associated with the best frequency that's being stimulated, the neurons being stimulated. These are called centripetal shifts. The other thing that happens is if you look at neurons that are further away from the best frequency of the stimulated cortical region, their tuning properties now shift away. So you're getting two things happening here, well, three things simultaneously. You're getting an enhanced response at those neurons that like to fire to the same frequency that the cortical neurons like to fire. Neurons that are close by, in terms of best frequency, have now shifted their tuning curves towards the frequency of the stimulated cortical region. Neurons that are not close to that best frequency have shifted their tuning away. Essentially, you've enhanced the contrast there at the level of subcortex to now overrepresent sounds that are close to the stimulated cortical region. Now, given that these cortical fugal projections appear to be a common feature of the auditory system in many mammals, I've just shown you big brown bat, but they're also there in rat, they're also there in cat, we can speculate that similar processes are at work in human listeners. And of course, in humans, we're not afforded the opportunity, at least not often, to take recordings directly from the brain at the intracellular level. So we have to look at indirect measures, and the best indirect measure that we have to date, I would argue, for looking at this kind of system is the auditory evoked potential. So what these potentials represent is the firing of a whole group of neurons that tend to be oriented in the same direction. So when neurons fire or when neurons communicate with one another, there's an influx of sodium in the basal end. There's an efflux of potassium on the other end. This creates a dipole, not unlike a battery. And if you have enough of these neurons that fire at the same time, you have a very large dipole, and the voltage difference here between these two ends can propagate through the neural tissue to the scalp, where we can record it with electroencephalographic electrodes. That's the EEG signal. By itself, the EEG signal won't tell you too much. It's going to look like this, very noisy. You can't tell what waveforms or fluctuations in here are related to auditory processing at all. There's just too much going on in the brain. So we need to isolate those particular voltage fluctuations that are specifically related to the processing of the sound. And the most typical way that we do this is we present sound repeatedly. For example, here, dog, 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 dog. We insert time markers into our EEG trace, and then we cut the EEG trace around that time marker, and we average it together. And this averaging process effectively attenuates all of that random neural activity that isn't directly related to processing of the sound, but it leaves or it allows for the summation of that activity which is directly related to the sound. And this is what I'm referring to when I talk about the auditory evoked potential. Now, one AEP that's become very popular recently is the so-called complex auditory brainstem response. And much of its popularity is attributed to the work of Nina Kraus, who will be speaking tomorrow, and some of her colleagues. This response reflects the phase-locked activity in rostral brainstem, namely inferior colliculus. This is why it's popular. As I said, the IC is a convergence hub for bottom-up projections and top-down projections. So if we're going to see modulation of subcortical activity, we might see it here. Now, the CABR has two components, a transient component and a steady-state component. So the transient responds to transients in the acoustic waveform, the steady-state to steady-state components in the acoustic waveform. And when we just talk about the steady-state response, which is what I'll be focusing on most in my talk, it's also known as the FFR, or frequency following response. And this response is capable of representing signal periodicities up to about 1500 hertz and its major generators are thought to lie directly in IC. That assertion is made based on work in cats, where if you record EEG signal before and after cooling subcortical cooling of inferior colliculus, you see that this cooling dramatically reduces the amplitude of the FFR at the scalp. I said that the complex auditory brainstem response can represent periodicity in the signal, and I'd like to point out here that this is actually true of both the signal envelope and the temporal fine structure. So the CABR is actually a combination of these two, and the reason for the reason for this is that envelope extraction happens very early on through the nonlinearity of the hair cell. So you can actually take a look at encoding of both of these two things by looking at the CABR, and you can actually enhance the representation of one or the other through particular recording techniques. I won't get into that now, but it's just important to know that both of these things can be represented. So now with the CABR, we have a tool for looking at brainstem encoding that we might not have otherwise had. There are many tools or many AEPs that are thought to reflect auditory cortex, and I'll get activity in auditory cortex, especially as it pertains to prediction or expectancy, and I will get into those. But now we can see if those components are related to encoding at the brainstem. So this experiment here, let's see if the CABR is indeed at least affected by expectancy and prediction, as you might predict or expect from that model of dynamic control. And it does appear to be. So Nina Kraus and colleagues in 2009 ran this experiment here with two sequences of the syllable sound da while recording the CABR. And in one of the sequences, or I should actually first point out that the da syllable has a form transition at the beginning of the voiced alveolar stop as the D transitions into the steady state vowel sound. And in one sequence, this transition was predictable here. It was repetitive. And the other one, this was modified. When we take a look at and compare the CABR response recorded by the repetitive to the variable condition, we see that the repetitive condition shows an enhancement of the second formant here relative to the variable condition. And you can see that here too for harmonic 2 and harmonic 4. Moreover, the normalized difference in the magnitude of the CABR, especially at the second formant here. So the difference in the representation of this signal at supposedly IC, when the signal is repetitive versus when it was variable, correlates really nicely with normal hearing listeners' speech in noise performance. So just to summarize up to this point so far, I would argue that listening can no longer be accurately modeled as a purely passive sensory process. Ascending bottom up and top-down descending projections work in dynamic loops to modulate the input of auditory information. For studying such systems in humans, it appears that the CABR or FFR, insofar as it reflects activity at inferior colliculus, is sensitive to this top-down modulation. And finally, the degree to which the CABR or FFR exhibits modulation is related to or is able to predict people's speech and noise performance. Okay. So I know that was a lot of information so far, probably some information that you're not all used to hearing. So if there are any questions at this point, I'd be happy to answer them before we move on and I talk a little bit about my doctoral work. No? Okay. So my doctoral work is focused mostly on the concurrent measurement of subcortical and cortical AEPs. So I've shown you a lot of evidence to say that we have a way of measuring this. What I haven't shown you is the evidence that we have for measuring this. I'll get into that. But my work was basically trying to bridge these two things to show the existence of this. So let's talk a little bit about encoding in auditory cortex. Now the cortical AAPs that I'm going to talk to you today about are a little different than that subcortical CABR. The subcortical response follows the periodicity in the signal. Cortical responses don't tend to do that, save for like the 40 hertz auditory steady state. Instead, they're transient, so they only occur once following stimulus presentation. And they're usually defined by their amplitude at a particular electrode and their latency relative to stimulus onset, which is this line here. That leads to a particular naming schema where, say, the first negative going potential will be named N1, the first positive going potential will be named, well, this would be P1 here, this is P2. I've highlighted this because this is referred to as the N1 P2 complex, and it's thought to reflect an indexing it's thought to index the integration of exogenous stimulus features at the level of auditory cortex. And the N1 further is particularly interesting because it may be a possible correlate of listening effort. So the N1P2 gives us a pretty good picture, or it's thought to give us a pretty good picture of very early level processing by auditory cortex. But it doesn't tell us too much about how auditory cortex might encode sound features in a way that helps it process change or detect auditory change. For that, we look at a different class of cortical potentials. And these, unlike the N1 and P2, are not elicited every time you hear a sound. They're elicited in very specific contexts. So the mismatch negativity is sensitive to the degree of deviance in an ongoing sound stream. The way we typically look at this response is we present a series of frequently repeating sounds. These are identical, spectrotemporally identical to each other. They come from the same location. They're completely alike. And then infrequently and at random, maybe 10% of the trials of the recording, we change one or more features of the sound. So we record and average the response to all of these, and we record and average the response to all of these, and we compare the two, and the mismatch negativity lies in the difference between the standard and evoked response. It's an additional negative response that you get whenever the auditory system detects a change. Now, this change detection process isn't simply related to adaptation or what we refer to as stimulus-specific adaptation. That is the fatigue of neural responses when they're presented with the same sound over and over. And we know that because you can elicit MMN even when the sound is constantly changing from trial to trial. And that is, MMN is sensitive to violations of acoustic trends, not just particular acoustic features. So you could present shepherd tones. These are tones that sound like they're ascending continuously in like an ongoing staircase. And you can violate one of the tones that would be essential in that pattern, and you can get MMN. You can play a melody, a sequence of notes, with one note out of key sometimes, and you can elicit MMN. So this is not just related to the indexing of spectrotemporal features, but it's thought to involve auditory memory processes. P3A is similar to the MMN, but it seems to require more auditory attention. So whereas the MMN can be elicited even when the subject isn't attending to the sound stream, they should be awake, but they don't have to be attending. They could be doing a different task. The P3A is only elicited if you have the subject focusing on the auditory task or at least not distracted by a secondary or tertiary task. So you could have an ongoing paradigm similar to this and have the subject actually detecting flashes of light, and you'll still get MMN. But if you play that similar sequence like this and have them detecting flashes of light, you'll no longer see P3A. So this is thought to reflect attention-orienting mechanisms that are triggered by the acoustic novelty. And it's also been implicated, its morphology, that is, its amplitude and latency, has been thought to reflect listening effort. So my work in bridging these responses has used an amplitude modulated signal. You can just think of it as a pure tone for this. The amplitude modulation is not important. But the signal this tone was presented in a relatively unique paradigm called the forward and reverse oddball. And the forward and reverse oddball, you present one sound, say a tone at 300 hertz repetitively, and a different tone infrequently and at random, say 700 hertz, in the forward presentation block. Then you take the same subject and you present them with the sequence where the actual assignment of standard and deviant tones is reversed. So now this lets you take a look at the change detection responses that happen when you present an oddball tone. And you can take a look at those responses when they're elicited by the same physical stimulus. So now you're not confounding your results by differences in the actual acoustical features of the sound. This is just what happens when the system detects change, period. So these are the stimuli I used. As I said, they're amplitude modulated pure tones. For anybody in the audience that may be interested, I used amplitude modulated tones so that I can also evoke auditory steady state responses and take a look at how those might be related. If you're interested in that, we can talk about it after. But for the purpose of this talk, we can treat these as pure tones. So here you can see the components that I talked about elicited by a 300 hertz carrier signal and a 700 hertz carrier signal. The standard component is in red, so this is the response to the repetitive stimulation. I only average those standard presentations that occur directly before an oddball. So this is not because of a difference in the sheer number of trials that went into an average. This pretty much, the standard presentation pretty much represents the most prediction the auditory system can experience, because it's the one that happens directly before a deviant presentation. Here in red, you can see the deviant component, and here in the dotted line is the difference. And in both of these cases, you can clearly see a P1, N1, P2, and you can clearly see a mismatch negativity response and a P3A. If we take a look at the same signal now, it's just filtered a little differently. So this is the same signal that I'm showing here. But now, rather than filtering this at between 1 and 20 hertz to visualize these slower cortical potentials, I don't filter the signal at all, and I look a little bit higher in the frequency region. We see 300 hertz represented nicely here for both the standard and deviant response. This is the response in the time domain. This is it in the spectral domain. And we can see the same thing is true for the response to the 700 hertz sound. And moreover, and more interestingly, in my opinion, when we take a look at how the energy is represented in this frequency following response, so this is now a spectrogram plot. If you're not familiar with these, they show time along the x-axis and frequency along the y-axis, and the z-axis here is the intensity of energy. You see, the representation of the signal when it's a standard is greatly attenuated compared to when it's a deviant. And moreover, this presentation as a deviant leads to an enhancement, a significant enhancement as measured compared to the standard response in a period of time that immediately follows these change detection responses. And it's the degree of difference between the standard and deviant response is actually related to the timing of these cortical change detection responses. We can see that that's even stronger when you just look at subjects with the strongest differences between standard and deviant invoked FFR. Moreover, subjects that exhibit strong representations of the FFR, and that's measured by cross-correlating the frequency following response with the evoking stimulus, show reduced latency of the N1. And if you'll remember, that's the cortical component that I suggested relates to listening effort. So, at this point, I hope I've convinced you that not only do dynamic control loops likely exist in the human brain, but that we can actually measure the action of these systems using auditory evoked potentials. but no matter how nice a hammer is, it's of little use without a nail. So the question really becomes, how might these sorts of measurements better inform us about the auditory system of listeners with hearing loss? And one answer to this question might come to us in examining how the system might be perturbed, especially when it comes to the representation of sound at subcortex. So here we see the temporal information represented for a normal hearing listener and a hearing impaired listener. These are actually groups of listeners, so this is an average response. This is the spectral response of the CABR. So that is if you took the time domain and you broke it up into frequencies. So these are the different harmonics that are in the syllable da. And here we see the representation of energy for the temporal envelope of the sound. When presented in quiet, there doesn't seem to be too much difference between normal hearing listeners and hearing impaired listeners in the temporal envelope, nor does there seem to be much of a difference in the temporal fine structure. So now we're looking at the actual spectral content and not the envelope here. But you add noise and something funny happens. Suddenly the hearing impaired group shows an exaggeration of the temporal envelope at these lower harmonics of the da syllable. That exaggeration isn't matched by a concurrent modulation of the temporal fine structure. In quiet, this difference is not too bad. Here what we see is the difference between the envelope and the temporal fine structure for normal hearing. It's quite low. For the hearing impaired group, unamplified, it's quite big. Amplified, it's a little bigger. You add noise, now this might actually become a problem, and it's a problem that's not overcome by normal strategies of amplification, but instead exaggerated. So now you see an increased overrepresentation of the temporal envelope at these harmonics here, and it may be why some clinicians, anecdotally at least, report that their patients in noisy situations remove their hearing aids because they claim that it actually helps them hear speech signals better. As I showed you before, the representation of the signal at subcortex in the FFR seems to be correlated with our cortical measures of listening effort. And if we just take a look at older and younger adults, now these are people with normal audiometric thresholds, all below 25 dBHL, we see that the older adults show an increase of N1 latency, similar to how we see here. And I don't have the slide to show you, but they also tend to show poorer stimulus representation at the subcortex. I can assure you that the situation isn't necessarily hopeless. I don't want to end on that poor note. You've heard my colleagues today, Eric and Petri, talk about the advantages of auditory experience and the advantage particularly that auditory training can have on the perception of sounds and maybe speech. And indeed, there is some evidence that such training can have an impact in the way that auditory cortex and subcortex act upon incoming sound. So here we see the CABR or FFR response elicited in non-musicians shown in blue and in musicians shown in red in response to a speech stimulus. And you'll notice that the brainstem, here's the brainstem response amplitude is reduced in musicians compared to non-musicians. These are older subjects with some amount of hearing loss, and we see also a concurrent decrease in the N1 latency in the musician group. Okay, coming to the final summary here, I would say that if you should take something away from this, in addition to the interim summary, it's that peripheral hearing loss might lead to changes in the way that how top-down modulation works, that increases the representation of temporal envelope relative to temporal fine structure, especially in noisy situations. And this might require then increased listening effort from cortex since it's not getting the signals that it needs to accurately encode the sound. I would also say that I think concurrent measurement of subcortical and cortical AEPs can be used to delineate how brain regions interact during different listening situations, and that such approaches might help us to better characterize how hearing loss affects different individuals and be useful in developing interventions and corrective amplification that's appropriately tailored to the needs of patients. Finally, as I said, the situation isn't hopeless. Auditory training might also be very useful in reestablishing the efficient processing and communication between subcortical and cortical structures. So, thank you for listening.